Hi, graduate students. Welcome to West Virginia University. We are all very excited and can't wait to help you with your awesome journey. My name is Martin Hruska, and I'm an assistant professor in the Department of Neuroscience. My lab studies synapses, which are the connections between brain cells. We are very interested to understand how synapses form during the development, how complex biology of synapses allows us to form memories and learn from experiences, and how defects in these processes lead to neurological disorders like Alzheimer's disease. We use very sophisticated imaging approaches to investigate these events. As you can imagine, synapses are very small structures and need special tools to observe, and we need special tools to observe how they work. So, we use super resolution microscopy to study how proteins inside of these structures enable their function. We have made very cool discoveries using this technique that helps us understand how memories might be formed in our brains. We also use molecular biology so we can change how the proteins inside of these tiny structures regulate synaptic plasticity. We can make mutants or change protein expression by editing DNA using novel approaches like CRISPR. In addition, we generate a lot of molecular tools that allows us to manipulate synaptic function. So the current projects in the labs are to understand the molecular diversity of synapses in the brain. We have more synapses in our brains than there are stars in our galaxy. But how does each synapse know what to become? Second project that we have is to understand how synapses fail in Alzheimer's disease. Loss of synaptic connectivity is the strongest correlate of memory loss in Alzheimer's disease. Yet we still do not know how and when synapses begin to disappear in this disorder. Finally, we are engineering optical tools to understand functional synaptic connectivity. How do neurons know whom should their synaptic partners be? There are trillions of synapses in our brain, but only subset of them are functionally related. We want to discover who talks to who. Now, besides the cool science that we do, you're probably wondering what is it like to be in my lab? Well, our motto is work hard, play hard. We are a very dedicated group of scientists that care about our work. Therefore, we like to discuss our science and projects a lot with each other. I encourage my trainees to talk to other people about anything science related as much as possible. This is how we learn to communicate our science to others, but it takes a lot of practice. I also like to walk through the lab every day and speak briefly with everyone to see how they're doing and whether they have everything they need to succeed. We will also have weekly meetings where you will talk with me in one on one settings about your weekly progress, but also about challenges that you had that week. Together, we will come up with strategies on how you can overcome those challenges. I like to fix things as quickly as I can to get you on your way as soon as possible. We will also have weekly lab meetings where one lab member presents their work to the rest of the lab. These are real presentations on PowerPoint, just like you would do at a conference. This is where you will hone on your presentation skills, but also you will receive the necessary feedback from your peers about your project. My goal is to make you succeed in whatever you choose to do. I am very committed to helping you along your PhD journey so you can get any job you choose once you are done here. However, this requires hard work and scientific rigor, discussing science, and being committed to your work. Well, then please come join us if you think that studying how our brain cells talk to each other is cool.